Okay. Okay. Hi, everyone. I'm Jason Kelly. I'm one of the co-founders and the CEO here at Ginkgo Bioworks. Super excited to welcome you all here today. I'll mention briefly, just sort of what you'll see behind us. Ginkgo operates kind of like an Amazon Web Services for programming biology, for programming cells. And we have about 250,000 square feet of highly automated labs here in Boston. And so, we're fortunate to get to see a slice of one of them behind us here today. And companies outsource their cell programming to us. In other words, Bayer in agriculture, Roche in pharmaceuticals would ask Ginkgo, could you program a cell to do something new for me? Cool. Yeah, it is actually pretty cool. I agree. Thank you, Catherine. And so when IARPA asked us in 2018 to basically do the opposite, we were quite interested. So the ask was, if you found a piece of DNA out in nature, right, or a cell that showed up in the Capitol building, and you wanted to know, was that DNA engineered? Did someone like a Ginkgo, could someone have programmed that cell, or did it occur in nature naturally? And so, we're really fortunate to have David Markowitz, who is the program manager for Felix, here with us here today. I will get to you in just a minute. So to me, what IARPA is doing here is, remember, this was in 2018 they asked for this. You might imagine this is actually quite a relevant technology today. This is a great example of sort of the federal government living in the future, right? Coming out and saying, this is a technology, it does not have private sector demand in 2018. We don't even really quite know exactly when we're going to need it, but I sense we will need it in the future. And then fast forward to today, and I think it is quite a useful technology. And so we're big believers in that role for the federal government here at Ginkgo. In fact, a sister organization of IARPA, so ARPA-H, ARPA for Health, which was just announced by President Biden a few weeks back, that the new director of that was Renee Wegerson, who's actually previously headed up technology for us in our biosecurity business here at Ginkgo. So we're really thrilled to see sort of the long bets that the government is making in these areas. You're going to hear today about two, I'll simplistically describe them as radar systems for finding engineered DNA developed here at Ginkgo and by our friends at Draper Labs. I'm not going to steal the technical thunder on this, as you're going to get to hear from the scientists and engineers themselves, but it's really amazing technology. So you'll hear from Laura Seaman and Erin Rosenberger at Draper, and Josh Dunn, who headed up our technical effort here, and Matt McKnight, who leads our biosecurity business at Ginkgo. There was an opportunity, President Biden released an executive order on biomanufacturing and biosecurity just a few weeks ago, and had a chance to give some remarks at the White House. The ability to design DNA at scale, I think it will be the foundation for what I'll call the DNA age. Think like the electronics age was the last 50 years, except I think it's likely to be more important. I think engineering biology allows us to address challenges in areas like climate, in manufacturing, in food, in medicine, and more. But I think COVID has given us a real indication of sort of the power of biology to do harm. And so, today there is a strong international consensus against the weaponization of biology. This is fundamental. Human beings, we are made of biology. We are literally our walking biology. And biology also makes all of our food. All right, it produces the atmosphere. Okay, it cleans our water. Right? It is essential that we keep this technology from being misused. And we look to the governments of the world and leaders in our defense and intelligence communities to monitor and prevent such misuse. And so I'm hopeful that tools like Endar that you hear about today as they mature will help to monitor for bad actors and reduce the odds that engineered biology is misused. Lastly, I want to say that new technologies play an increasingly powerful role in our lives today. The technologies of designing DNA will be no different. And this is just a first step you'll hear about today in building out what should become a robust worldwide biosecurity network. The U.S. should lead here. And I'm proud to say today that I think we are actually leading in this area, thanks to the leadership of Dr. Catherine Marsh, the director at IARPA, who had the vision to move not just this technology, but many technologies of strategic relevance to this country forward. We're truly honored to have her here with us today. And so, Dr. Marsh, I'll pass the mic to you. Thank you.
Thank you. Uh-oh. Are you getting me?
Yeah,
all right,
yeah.
Good. All right. Well, thanks. I am here back in Massachusetts. I'm from here. It is delightful to be back, but really, as the director of IARPA, so really worried about next generation, high risk, high payoff programs that can change the course of where the intelligence community needs to go now and in the future, right? That's what we were stood up to do. And so I love the fact that in 2018, we're ahead of the game doing this on behalf of the IC. Bio is one of the biggest areas that we have to worry about because you don't understand intention, right? Good versus bad. How do you know? Right? I tell everybody I love to garden, so I'm a really, really avid gardener. And my vegetable garden 3 years ago, I had to lay fallow because I had some sort of a fungus. And everything I planted died. And that was just really depressing. And so how do you learn from that? Where did that come from? And what's the threat that that possesses or presents for the intelligence community? So understanding and knowing what it is that this protocol could potentially do for the intelligence community is where we have to make those investments, right? And we don't have any idea up front whether we are going to be successful. We put investment in those areas where we want to move the needle and we want to change the game for the community so that we can send things out, understand, and then get some learning back. Ultimately, we're investing in areas where the private sector is not going to invest because that's not the threat that they need, nor do they need to follow on behalf of the community. So when we stood up the Felix program, and David's going to talk much more in depth about that, it was really to be able to understand the background and the genetics and whether or not we could really get ahead of this, because we put people in harm's way all the time in the intelligence community, and we need to make sure that we understand that. We have a level of protocols and execution of what we do in our community that others that pose risks to us around the world do not have. And so by making this investment and the tools that are going forward, we really think that we've developed a revolutionary new capability. And I'm going to turn it over to David to go into the details of where that research is going to go. Okay? Thanks, David.
Thank you so much, Dr. Marsh. And thank you everyone who's joined us here in person and on the live stream. I'm David Markowitz, the Program Manager for Felix. I'm a biologist with a background in computational science and management training from MIT, just across the river from where we're meeting today. I think the founders of Ginkgo hail from MIT as well. So, and Draper was a spin-off from MIT, so we're very much keeping it in the family. So it's been my great privilege to lead this program during a period of history that underscores the pressing need to rapidly detect and understand emerging biological threats around the world. When IARPA launched the Felix program in 2017, methods for detecting engineering in biological systems were extremely limited and very labor-intensive. We only had the ability to do targeted searches for a small number of known modifications, and we could only do it in pure samples with DNA from one or just a few organisms. Furthermore, these tools were largely applied to the detection of genetically modified crops, not viruses or bacteria. The idea of creating a general-purpose platform for rapidly detecting genetic engineering in any type of biological sample, of any form of engineering, was literally science fiction when IARPA created the Felix program. Many people thought this was impossible because the problem that we had defined was like 1,000 times harder than anything that the capabilities at the time allowed you to do. And today we're here to show you that not only is this possible, but we have developed platforms that make general purpose engineering detection both routine and scalable. I want to emphasize, IARPA, the federal government, has independently confirmed the capabilities of the platforms that you'll be hearing about today. As Dr. Marsh said, the technologies the Felix program has developed are revolutionary and will greatly enhance the intelligence community's ability to quickly detect dangerous engineered organisms before they can cause significant harm. Furthermore, the tools we've developed through Felix will help to protect the trillion-dollar US bioeconomy by making it easier to detect theft of engineered organisms. This is the intellectual property that makes the bioeconomy run, and now we have additional tools needed to protect it. As with every IARPA program, developing the experimental and the computational tools needed for Felix to succeed It was a long, difficult, and multi-year process, and there was a very high risk of failure. Just to give you some more background about how we executed the program, Felix was architected with 2 phases. In phase 1, our research teams developed initial versions of their platforms, and IARPA tested and evaluated each team on a small number of organisms and engineering types. When I say tested, I mean we provided each team with a batch of biological samples with a composition that was unknown to the research teams in advance. And they had to report on the presence or absence of engineering in each sample. This pushed our teams well beyond the state of the art, but the testing samples that we used in phase 1 only captured a small sliver of real-world problems. Phase 2 was when we really pushed hard toward general-purpose detection capabilities. IARPA evaluated our platforms using a really expansive set of organisms that included viruses, bacteria, fungi, plants, and complex samples taken from soil and from mammals. We engineered these samples to represent all possible genetic modifications that one might encounter in the world, ranging from large insertions and deletions, which are relatively obvious, down to single base edits made with CRISPR. And we made the samples quite complex to mimic real-world conditions. We had mixtures of organisms, dilutions where the engineered organism was quite rare in the sample, and what are called metagenomic samples that mixed DNA from hundreds of different organisms together. Now that might sound like a nightmare scenario, but it reflects the real-world challenges faced by national security stakeholders every day.
And we knew the ground truth, right?
We knew the ground truth because we had engineered the samples ourselves. So the challenge that the national security community faces, both the intelligence community, DoD, and our partners around the world, is that we simply never know what sample is going to come through the door in a government lab. And we need to be prepared for anything. This is the kind of ambitious, multidisciplinary problem that IARPA specializes in solving. We do moonshots. And we succeed by investing the necessary resources, by actively managing the research, supported by teams of government scientists, and by holding everyone on the program accountable for success through independent testing and evaluation. Dr. Marsh and I were confident of the program's chances of success because of IARPA's incredible partners that we had in developing these technologies. Felix supported work by 6 teams of contractors working on experimental and computational platforms. These teams were led by principal investigators from Draper Laboratory and Ginkgo Bioworks, who we are joined by today, as well as by the Broad Institute, by the Wyss Institute at Harvard, by Noblis, and by Raytheon BBN. When IARPA announced the Felix program in 2017, we set the goal of demonstrating platforms that can detect 90% of engineering modifications across a panel of samples. That was extremely ambitious for 2017, 90%, considering that there was nothing in existence for general engineering detection at the time. But since 2017, the problems have gotten significantly harder, even than we imagined back then. Biologists have gained the ability to make very small genetic edits in basically any system. And at the same time, the complexity of samples that are of greatest interest to the national security community has increased dramatically. If you have ever checked the internet for the latest COVID wastewater monitoring data, that's characteristic of the type of complex samples that we have to deal with now, where you have everything but the kitchen sink mixed together in one sample. When IARPA conducted our final evaluation of the Felix platforms earlier this year, we tested them on the full spectrum of current biotechnology, biodetection challenges as they exist in 2022. And I'm happy to say our teams performed amazingly well on these real-world evaluations. Today we will hear from the Draper and the Ginkgo teams whose experimental and computational platforms performed most impressively on our evaluations. In both cases, they correctly detected about 70% of engineering in a panel of 100 complex samples and had no more than 1 false positive. I think Draper had 0 false positives. It's truly amazing. Now, as I said before, this was science fiction when Felix started, and now we have automated platforms that rapidly and sensitively detect engineering in almost any biological sample.
How rapid is rapid?
It depends on the sample. If it's a virus, you can do it in 8 minutes. If it's a plant genome or a mammalian genome, it can take several hours.
Okay, but still, that's better than weeks and months.
Correct. It used to be a very labor-intensive process that could take months, depending on the complexity of the sample. So we are here today to celebrate the individual achievements of the teams who made this possible, as much as the Felix program's overall success. And with that, I'm happy to give the floor to colleagues from Draper Laboratory, to Laura Seaman and Erin Rosenberger, and then we will hear from our colleagues at Ginkgo.
Thanks, David. So I'm going to talk to you today a little bit about some of the technology we developed at Draper on this program. Next slide. Okay, and specifically, we worked on doing a hybridization-based approach to this problem. Next slide. I guess start with introductions. I'm Laura Seaman. I'm a principal scientist at Draper and the machine intelligence group leader, and I was the bioinformatics task lead on this program. Erin, who will speak in a minute, is a senior member of the technical staff in the biological microsystems group and was the assay development lead. Next slide. So at Draper, we are an independent, non-for-profit engineering innovation company working on a wide variety of challenging and important problems. We have over 1,700 employees and work on a wide variety of problems, including, most relevant today, a lot of different biological systems, including the work that we did here today. Next slide. Obviously, we've been talking about why understanding genetic engineering is important, but this is a little bit about how we tend to think about that problem. So genetic engineering has been happening for quite a while now, and it's become increasingly easy to do it. So whether we're talking about mice being intentionally released to help reduce Lyme disease, or DIY gene editing kits that you can order on the internet and anyone can have, it's important to be able to understand how these tools are being used and be able to identify them in an unknown situation. And in particular, our effort really focused on dilute and complex samples, where there might be lots of different things in there and only some small fraction of it is engineered. So these real-world samples could come from a sink trap or from soil. If you're needing to find engineering in that, you have to wade through all of this genetic material that isn't engineered to find the small pieces that are. And so we partnered with collaborators at Harvard and BU to do this work. Next slide. So our approach was called TEMSeq, which stands for Targeted Engineering Marker Sequencing. This is a series of lab methods to capture and enrich DNA sequences using DNA probes. So essentially what we're showing here is on the left, you can have a complex simple sample, in this case something like soil, and the blue and orange pieces in there represent DNA that's engineered, where you've got an engineering marker in the middle with some kind of normal DNA around it. And so these engineering markers might be things people commonly use for engineering, or and the tools they use, things that are going to be left behind when that engineering happens. And we use DNA-based probes affixed to a surface to capture that DNA, so that the DNA in the sample will bind to the DNA that we're looking for and enrich it so that we can wash away all the DNA that isn't relevant, that doesn't have the engineering, and focus just on the DNA that does have that engineering. As part of this process, we also built a bioinformatic pipeline to help with the interpretation and understanding of these results. In addition to a fluorescence output and imaging-based output, most of the time we ended up working with sequencing data, and that's really kind of complicated to deal with and understand. And as Erin will talk more about in a minute, we also built a mini microarray that we call a chiplet, which is a piece of hardware that we use to analyze this, especially in a case where you have a precious sample, you don't have much material. Next slide. So essentially the way this works is that we focus on these engineered regions. And this is showing an example of the data that we collected on the program, where we had an engineered region in an organism, in this case P. aeruginosa. That's what the blue represents, and then that red region is the engineered. And in this case, we were looking at a dilution. So there's just a little bit of engineering mixed in with a bunch of the normal sample, and that's why in the top row there, you see a lot of DNA from the organism, but you don't see that engineered region. Because it was dilute, it just doesn't come up very often. But when we do our capture, you can see that that's when you can see the reads in that engineered region, and that's what allows us to identify that this has in fact been engineered. Okay, next slide. As part of this, we had to build the computational tools to detect this engineering. On that previous slide, I was showing something where we knew what was in the sample. We had done the engineering internally for testing, but most of the time we knew our goal was to look when we didn't know what it was. So it's important to be able to start with just the engineered region and figure out the context from there. And so that's where we built these automated processing pipelines to make sense of this data. And what I'm showing here is an example of that output. Along the top is the read depth, and you see the peak over the green engineering markers because that's where the engineering markers are. That's what we were capturing. So we get a lot of information from that area as well as some from all of the surrounding regions. And then we're able to use assembly and other algorithms to kind of build up that context and understand what was put in, where it was put in, and even potentially get some understanding of what it might have been designed for. And so we had to build all of these automated tools, build some custom algorithms to deal with differences in our data versus kind of what these algorithms are normally built to deal with, and make sense of the sequencing results. And create these visualizations so that an expert can quickly review the system and say, okay, yes, this is clearly engineered, and understand how that's been done.
Does it rely on an expert to be able to make that?
We have an automated caller. We have found that experts still tend to be better than our automated system. So we have some of each available. Next slide. And I will pass it over to Erin.
Thanks, Laura. Yeah, so as Laura mentioned, as part of this program, we developed a custom hardware that we call the chiplet, or mini microarray. And basically, as Laura mentioned, we use DNA probes to really enrich the sample for what we're interested in looking at. And in this case, we immobilize those probes onto a small glass substrate. And it works similar to a traditional DNA microarray, which uses an array of DNA spots that can capture nucleic acids from a sample. And also similar to a traditional microarray, our mini microarray provides a optical output for quick identification of what may or may not be in the sample. However, we have some added benefits that are specific to this mini microarray platform. First of all, it's very small, and I will do a demo in a minute and kind of show and show you folks at home as well. But it fits into a standard 1.5 milliliter microcentrifuge tube, which is traditional for biology labs. And the way we designed it, it requires less than 10 microliters of sample. So you can imagine that's maybe half the size of a eye drop that you can imagine putting into your eye. So very, very small. We also designed it so we can recover that sample. So it's actually a non-destructive way to analyze these samples. So you can recover that unenriched sample and process it elsewhere. So we thought that this was really important for the intelligence community where some of these samples are very precious, there's very small volumes, and you want to do a ton of testing. So you don't want to waste large volumes of sample. We also demonstrated that these prepared glass substrates are shelf stable for at least 6 months when stored at room temperature. So I think I will stand up to do the demo. If you want to go to the next slide, please. Thank you. So I'm going to demonstrate this with just some red water, but you can imagine that this is your highly complex sample. So a lot of genetic information, and you're looking for one very small component to that.
This is something you collected in the field, right?
Exactly.
From wastewater or anything around the world.
Exactly, yep.
You can analyze it at the point of collection.
Yep. So you can, there are some processing required to get the nucleic acids, but we actually are working on a program, and this was designed to be fieldable, so we are working on developing those protocols to be even more fieldable so you can do it directly at the spot. So first I will pipette the small volume.
You need an extra hand.
That would be great. Thank you, Laura. Yeah. So I'm just grabbing the small chiplet right here. It's about the size of a thumbnail. And now I'm pipetting the small sample fluid into the chamber. So again, red water to just demonstrate.
And if you go to the next slide, there are images also.
And I didn't bring a bio-waste container, but it's not bio-waste. Yep, just water. Oops. And so now the sample is loaded into the chamber of the microarray. And at this point in time, the sample, the fragments of DNA that, that contain the sequences of interest are being captured onto the glass substrate. After the incubation, we can just do a quick spin in this is just a battery-powered microcentrifuge tube. And that allows us to recover that sample. And so you can see now that the recovered sample is collected at the bottom of the microcentrifuge tube. And you can also see the chiplet sitting directly in that microcentrifuge tube. So at this point in time, this recovered sample that's sitting at the bottom can then be used for additional processing. And we did show, we were able to test it on a different microarray with a different set of probes. And what is collected on the chiplet surface is really just those sequences that we're interested in. So we're able to wash away all of that background, all of that extra genetic material. And if this was fluorescently labeled, you can image it just with a fluorescent scanner to get that quick kind of identification of, yes, this is present. But as Laura mentioned, a lot of what we were doing was sequencing, so now we need to actually recover that enriched sample. Can I have you hold this again? Thank you. So again, just blue water for this case, which is just our solution that allows us to melt away what's been captured onto that DNA surface.
High pressure,
Yes. Not used to this. So now again, a quick incubation just to melt away or dehybridize those captured DNA fragments. And another quick spin in our microcentrifuge tube. Takes about 2 seconds. And now what we have here in blue is our enriched sample. And so now this sample can then go into sequencing and run through the bioinformatics pipeline to give us more information about that really rare organism that was present in our sample. So if you go to the next slide. So yeah, just to briefly summarize, we developed a platform that allows us to enrich samples for detection of really rare organisms in highly complex samples. I think we were able to demonstrate down to 1 to 1 million dilution. So you can imagine, a million other organisms present in the sample, and we were able to identify that one genetically engineered organism. It also requires no prior knowledge of the organisms that are present because we are using the known markers of engineering. It could be any organism that's been engineered. Our custom bioinformatics pipeline really helped automate the system and simplify our analysis. And this custom hardware really is great for precious samples and is, can be fieldable. And as I mentioned, this technology, we developed it to be agnostic so we can kind of expand it beyond just the genetic analysis tool. And we're using it for other applications, one including a program looking at coral reef restoration. And that's where we've developed the fieldable optical detection component to this system. And we're also starting a program looking at wastewater surveillance, as was mentioned earlier. So next slide.
So true dual use, right? It's not just IC, right? It's true dual use applications, right? So we take this, right, use that we have for the intelligence community, and it's a much broader impact to that, right? And that's what the goodness of this kind of research is we're looking forward, and then they're taking it and doing a business case to develop other use cases that matter for the community writ large, the greater community. So I think it's huge, that you found these other ways to use this, and it's going to make a difference both from a climate change perspective and other areas of research that can benefit from the technology you've developed. So.
Yeah, no, we're definitely excited to kind of use this beyond, and just want to take a second to acknowledge that this work was done by a large group of researchers led by Kirsty McFarland and with our collaborators at Harvard Medical School and Boston University. So now I'll pass it to our friends at Ginkgo.
Thank you very much. Alrighty, well, hello everyone and good morning. My name is Joshua Dunn. I'm the head of design at Ginkgo Bioworks, and I served as our principal investigator for the Felix program. Today I'd like to tell you about a computational system we developed that detects genetic engineering in the wild. And we call the system Endar, which stands for Engineered Nucleotide Detection and Ranking. Next slide, please. So today I'm going to walk you through how Endar works in about a 3-step process. So the first step, which starts on the left, is sample collection. So you as a user would have some sample that you would want to assess for bioengineering and understand the nature of the bioengineering in that sample. So you would need to sequence the DNA from that sample, put it on a sequencer, and upload the sequencing data to Endar. The second step, shown in green in the middle, is actually running Endar itself. So Endar will execute a variety of computational analyses that help make sense of the sample and understand what is in there, detect the engineering, and then put that engineering in context for an analyst to really understand. And that moves into the third step, which is review of the outputs. And so an analyst will examine the outputs of Endar really to answer 4 questions. First and foremost, was the sample engineered? Yes, no, or do we need to take a deeper look? If the sample's engineered, where? So what genome in the sample was engineered, and where in the genome did the engineering occur? Third, how was the sample engineered? Was DNA inserted, removed, or replaced? Did that DNA have any genes in it, and where did the DNA come from? And finally, how or why, excuse me. So in addition to labeling the likely sites of engineering, we provide a lot of context in the Endar outputs that give trained analysts the ability to interpret what's going on, infer what the engineering might be doing, and make decisions accordingly. And this is actually really important because if you can understand the implications of an engineering event, you can select the right response. Next slide. Before we get into the guts of the system, I wanted to tell you a little bit about 4 design considerations that went into the building of the system. So first and foremost, we wanted to create a system that would not only tell you whether a sample was engineered, but also where and how to give that decision support capability I was mentioning just a second ago. Second, and this is really important, genetic engineering and biosecurity are in an arms race. Right? So as genetic engineering technology becomes more sophisticated and more accessible, tools like Endar need to keep pace. And so we made a lot of very thoughtful decisions in how we designed Endar to enable it to grow for the future, as opposed to relying on methods of genetic engineering today. Third, and related to this arms race and the proliferation of technology, we thought it to be very important to design Endar such that we could pull the tools inside of it out and repurpose them for novel question answering, such as threat detection. And the reason for this is that while we understand the important biosecurity questions today, we know new questions are going to arise in the future. For example, during the middle of this program, the COVID pandemic created a really dramatic need for analysis, not just of DNA material, but also geography, demography, and all these things had to be tied together. And finally, our system includes, at the end, as a pretty fundamental step, review by a human analyst. And the reason for this is that some very consequential decisions can be made on the basis of Endar output. So we want analysts and decision makers alike to be informed, intentional, and very confident in the calls that they're making. Next slide. So let's look under the hood a little bit into the 3-step process I mentioned, and we'll start with sample collection. So next slide. So we designed Endar to be applicable to a broad variety of sample types, and there are basically 3 broad categories. So there are pure samples where you've got a vial of literally one thing. So this could be a clinical isolate, an environmental isolate, or a laboratory sample. You could actually have low complexity mixtures containing 2 or 3 organisms. So this might be like a sample of a clinical sample containing both host and pathogen DNA. And then finally, most complex of all are metagenomic samples. So these are samples that contain tens to thousands of species, such as you would see in wastewater sequencing or human microbiome samples or the like. So regardless of the sample type, what would happen is an analyst in the laboratory would extract the DNA, prepare the DNA for sequencing on a next-generation sequencer, and run the sequencing instrument. And what the sequencing instrument will do is it'll read the sample in the DNA, and it'll generate a large data file containing thousands to millions of short segments that each provide a tiny little view on some part of the genomes that were present in the sample at the beginning. And right now, Endar supports all the commonly available sequencing formats. So we support short read data coming from Illumina instruments, long read data coming from PacBio and Nanopore instruments, as well as hybrid samples containing both types of data. Next slide. So now let's get into using Endar. And as I mentioned, there are really kind of 3 conceptual steps that Endar performs. And the first is sense making. Then we detect engineering, and then we put it in context. So the first step in the pipeline, next slide, is the preprocessing module. And so this module takes all of those reads, reconstructs the genomes that were present in the original sample, and annotates those genomes with landmarks that helps an analyst later on understand what they're looking at. So this will annotate genes. It will annotate quality control areas, so areas of the genomes that are more likely to false positives, so that we can actually control that inline as part of the process, and a whole bunch of other types of information that place the engineering in context. After preprocessing, next slide, we move into the inferred reference module. So a lot of Endar's components work by comparing the DNA in the sample to panels of known natural genomes that we've seen before that we know are natural. And so what the inferred reference does is it looks at all the DNA in the sample, identifies the species, and then finds the genomes of the closest relatives of those species that we've ever seen before. And this lets us perform a lot of fine-grained comparisons later on. So after we've done those sense-making steps, we can move into engineering detection. And so Endar contains a bunch of different algorithms for detecting engineering, and this makes the system robust across scenarios and use cases. So the first and most simplest component is the key indicators module, and what this module does is it looks for smoking guns of engineering. And what we're defining as a smoking gun is a sequence that's pretty rare in nature, but very common in engineered materials. So these could be things like green fluorescent protein or antibiotic resistance markers or other genes that are commonly used in bioengineering. So right now, Endar has a database of about 8,000 such indicator sequences, both proteins and nucleic acids, that we can tailor to use cases and deployment scenarios of interest. One thing I would also note is that because, and this is a fundamental challenge of the program, because each of these sequences does arise in nature, if you're sampling something like wastewater, you're going to see this popping up all the time. So in addition to labeling these sequences, we actually give you a context score so that if you're seeing one of these indicators in the species or in a related species where you expect it, we can downweight that. Where if you're seeing a species clearly, an indicator clearly where it doesn't belong, it'll glow, bright red in the system. So the key indicators module is really good for finding smoking guns and obvious cases of engineering. But not all engineering is obvious, and not all engineering contains a smoking gun. So for that, we have another component called the prefilter, which is on the next slide. What the prefilter does is it takes the DNA in the sample and compares it to a large database of known natural reference genomes. And when it detects differences between those two, it'll use machine learning to infer whether these differences are really due to engineering or whether this is just a natural genome we haven't seen yet. And so this raises the accuracy of the entire system because we can label something that we believe is natural or label something that might just be a technical artifact and isn't actually real engineering. And then moving on to the junction localization component. So this component basically reverse engineers how the engineering happened on the next slide. So most genetic engineering occurs through cut and paste operations, kind of like what you do in a word processor. So what you'll actually do is cut and paste pieces of DNA from one genome and put them into another. So what this module does is it takes the sample's genome assemblies and all the references that we inferred were in there and infers the cut and paste operations that happened to make this genome occur. One thing that it can do even beyond that, which is great, next slide, is when DNA is inserted, if the DNA is coming from a known source, it will tell you that, and it will also look for all the genes that are in that inserted DNA. And furthermore, when DNA is excised from a sample, so when it's deleted, this module can actually call out to public databases, figure out what the missing DNA was, and if there are genes that were in that missing DNA, we can tell you now what genes are absent from the sample computationally. Next slide. So each of these components produces a ton of contextual information about all of the engineering events and everything weird in the sample, and examining that directly is actually pretty daunting. It's like drinking from a fire hose. So we have another machine learning enabled component called the ensemble, and what this does is it integrates all of the information from those upstream modules to perform two functions. First is it identifies all the regions of interest in a sample that an analyst should examine. And second is it applies a probability or a score to each of those, indicating the confidence we have that that region was engineered. So as an analyst, what you get is a rank-ordered list of places you should look at, along with the score or the likelihood that that region contains engineering. So moving on to step 3 is examining the output. So we have a reporting and a UI component that takes all this output and creates really two outputs. The first is a PDF document designed for an executive audience to get a good sense, high-level sense of what's in the sample. And the second output is all the detailed bioinformatic data files that result from all the complex analyses going on. So let's take a look at the executive report. Great. So here's a thumbnail of the executive report. And this report really describes a high-level determination of what is in the sample. If a determination can't be made, it includes a relative level of effort required to make a determination when an analyst looks deeper. And it includes a lot of quality control stats. So for example, data quality, what species were found, how closely related those species are to anything we've seen, how many regions of interest were called, their scores, and thumbnails of those actual regions. So what this report enables is a team lead or an executive to get a sense very quickly of what's going on, and as needed, triage a sample to the appropriate human analyst in their team. So looking deeper, we can take a look at the bioinformatic data files. So here I'm showing you an actual image of Endar. What we're looking at is data from one of the actual T&E testing and evaluation samples provided by IARPA. And this is the screen that an analyst would see when they're going into Endar. So the x-axis here is DNA, and if we zoomed in, you would see little A's, C's, T's, and G's. Here we're zoomed out. We're looking at an entire yeast chromosome, so about 750,000 base pairs of DNA. And you can see all those little colored boxes. Those are landmarks annotated by Endar. And the one that I want to call your attention to is the bright red one in the lower middle of the screen. What that is a high confidence region of interest called by the system. And so this is the only region of interest called on this chromosome that an analyst should look at. And an analyst looking at this would know to zoom in and click on that region right away. And so if we go to the next slide, this is what they see when they click in. And so moving from top to bottom, you can see at the top there's a pink region labeled insertion. So Endar has determined that some DNA has been added to this sample. Below that, we can actually see the structure of those insertions. So there's some gray segments of DNA. These all come from known sources, and a green segment of DNA that I'll talk about in a second that we couldn't figure out the source from. And if you click on those gray sources, what you actually get, that's the next slide, you'll get a mouse over telling you what species it came from. So this piece of DNA actually also came from Saccharomyces cerevisiae, specifically strain S288C. You also get the accession number of the sequence in NCBI, the coordinates of the DNA, where it came from in that sequence, and the NCBI taxonomy ID. So these are all bioinformatic hooks that you can plug into other pipelines to really understand what's going on. Now, back to the green sequence, the green labeled sequence, the one that we could not figure out the source of. We can actually tell what's going on by looking at the key indicators output. So down in bright green toward the lower end of the screen, if you have really good eyes, you'll see that unplaceable piece of DNA encodes green fluorescent protein. So green fluorescent protein is one of these common engineering markers in biology. It makes things glow green. We can additionally see that it's under the control of genetic markers under the control of regulatory elements that confer high expression on heterologous proteins in yeast. So a trained analyst would look at this and know pretty much immediately, hey, this strain of yeast is likely going to glow bright green. So this is just one of the types of examples of analyses that we can do with Endar today, where we can very quickly zoom in on a sample, see what's going on, see DNA was inserted, and even make pretty good guesses about what the function is. And so I'll hand it over to Matt to talk about how we're going from this to a bigger future.
Thanks, Josh. I'm Matt McKnight. I lead our biosecurity business here at Ginkgo. And just so everybody gets a sense of my daily life, it's mostly talking after people like Josh. So it's awesome to be here also with Draper, which is equally amazing work. And thanks to the IARPA team for joining us and giving us the opportunity to take and deploy the work that has been done here for over a decade against some of these big problems. So, I think what I want to do in transition to Q&A is just share a little bit about how we think about these capabilities and where to take them. I think the reality is, and it's something that we hold deep here at Ginkgo, is that modern biotechnology matters more when it is deployed at massive scale. And that is the transition from an amazing world of R&D and science into biotechnology being deployed in support on the economic side, in support of new ways of production, on the security side, in ways to keep us all safe from what we've seen for the last 3 years, or in the future, potential genetically engineered threats. And I think we should all look back and learn. The one major thing we should learn in this category from COVID was the story of our response was modern tools of biotechnology being deployed at massive scale. So everything from, if you look at just some of the work we've done, taking PCR technology, and many others, by the way, not just Ginkgo, but we know we're very close to it, taking PCR technology, essentially a lab technology, and deploying it to the tune of hundreds of millions of tests across the country. We run millions and millions of tests in K through 12 schools as a detector for outbreaks that you can then intervene with. Similarly, that program is being used in airports now to do genomic surveillance, monitoring when you have new variants, so you can get early warning of new variants, so you can respond with public health tools, with technology tools in a more nimble way. if you look at the mRNA vaccines, I think it's very similar, right? That is something that was R&D before COVID that turned into billions of doses delivered around the world. That's logistics, that's operations, that's biology not caring about borders, so we need to deploy these technologies globally if they're going to act as biosecurity technologies. And so from our standpoint, the exciting thing about what Draper's doing, what Josh is doing, specifically on our side with Endar, is how do we then take and quickly in our biosecurity function make these available to all of our programs that are doing large-scale collection across the world. And that is where you combine amazing technology development with scale distribution, and you get to proxies like thinking about collecting ATCs and Gs and monitoring them, DNA and RNA, and monitoring them like we think about cybersecurity. We collect the zeros and ones on the planet to monitor for malware constantly. And we're constantly with a mindset of improvement of product. So from our standpoint with Endar and Josh's team and otherwise, we want to keep deploying it globally and keep improving it so that, as Josh mentioned, when we design it for future threats, it's continually a mindset of, okay, how are we upgrading and deploying rapidly, just like you do in the software ecosystem. So we care really deeply. We're really excited about the partnership with government. Like, this is not a private sector to solve all problem. This truly needs to be a partnership. Government has this role to protect citizens around the world. We just want to continue to be good partners, and thank you for the opportunity to do that. I will turn it to any Q&A that might exist. Riley?
Oh.
Very advanced.
Hello, can you hear me? Hi, my name is Riley Griffin. I'm a reporter with Bloomberg. I cover biosecurity now. And I have a question for you, Dr. Marsh. As IARPA looks at the culmination of the Felix program and all the learnings from these various projects, I wonder how you're thinking about advancing the conversation to get not just to the genetically engineered detection, but attribution, and what IARPA can do to close the gap, not just finding something that is genetically engineered, but advancing the national security objective and of better understanding who is doing that engineering.
B24IC. I mean, so we have a program on the street right now targeting bioinformation and biosecurity, right? How do we know who's doing what and where, and how do we protect against it? And so we literally have just received a large number of proposals that we are sorting through to decide who will be awarded to go down that field, because and it is what we call a targeted seedling effort, so it's a shorter effort with the intent. As you're alluding to, the field is so broad right now, we don't know specifically what to go after, and so we don't have enough to do a targeted program, and that's what this, what we call a super seedling, is going to give us. It's going to give us that framework, which we hope will turn into a program. So while this is a 2-year dedicated effort on probably 12 to 15 different research approaches, we'll see whether or not 2 or 3 approaches result in what that follow-on program will be to get after that intent, right? Because that's what we care. It's not the only thing we care about, but it's certainly one of the most important things from that being able to know early, right? Because it's not just intent, it's got to know early before we can have an impact from it, so we can do deterrence, right? So that's what I have, I think.
I think one thing we've seen with, say, cyber, or even if you look at like satellites, is getting to a baseline. What does the world look like when someone's not trying to do something, right? And we have that when it comes to satellites. We have that when it comes to our computer networks. We do not have that when it comes to viruses. We have a very spotty monitoring network that's sort of kind of been built up on the back of public health, that a lot of which was not well supported. In particular, I would say globally. And so we're missing that awareness of a baseline, and without the baseline, it's hard to know where something popped up. So I think that's another place, and this is very much a thing that would benefit both the public, private sectors on national security, private sector from a standpoint of there's a lot of knowing what's happening in the world genetically actually is a lot of assets for the biotechnology industry. And then third, public health. Like, let's not forget, nature throws this stuff up all the time, right? So actually having that monitoring, I think, will also help us just from a global public health standpoint too. But without it, I think it's hard. Yeah.
Thank you. I have one follow-up question for Dr. Markowitz. Wanted to ask you, you mentioned in passing the applications of this kind of technology to protect intellectual property. And I wanted you to expound on that for a moment or two, because I think the national security risk is obvious, but perhaps the private sector risk is something my readers are less aware of.
It's not something that typically catches headlines, detecting intellectual property theft. But when we're talking about protecting an emerging trillion-dollar segment of the U.S. economy, you can measure the impact of this program in potentially billions of dollars of economic value protected. The applications to detecting IP theft are if you are sampling at ports of entry and exit from the United States, right? Airports, shipping ports, things like that. If you're collecting samples there and you want to be able to rapidly detect if somebody's trying to smuggle something out of the country, Felix tools empower you to do that now, to say this, I know what this is, I've seen this before, this is engineered and it matches a database of IP, right? And so US law enforcement theoretically can use Felix tools to implement that capability now. If you have legal disagreements between companies that are competing in the bioeconomy and you want to determine who is using whose intellectual property, we now have a straightforward, rapid means of answering that question. And that's a tool that there wasn't as significant a need for it before Felix, but now we have it. So it's not as eye-catching an application as stopping bioterror, which you measure the impact in lives saved. And one thing I want to emphasize is that most of the time when the intelligence community is doing its job, you don't hear about it because nothing bad happened. And that's the value that the Felix program is going to deliver. If we have deployed these tools, we have transitioned our computational and experimental tools to double-digit numbers already of government laboratories in the intelligence community and the Department of Defense, as well as many international partners who already have these capabilities in-house and are evaluating them for deployment. Once the true impact of Felix is realized, you're not going to hear about it anymore because it's going to keep all of us safe. It's going to protect my family, your family, and the value of the future companies that are driving the U.S. bioeconomy. So hopefully that answers your question.
Thank you.
Thanks, and we want to remind folks to please ask questions in the YouTube chat. I had a question that came in online for Draper. And so they're curious about what the chiplet offers above and beyond regular target enrichment protocols.
Yeah, so I think the main thing that I'll kind of reiterate on the mini microarray is the ability to process very small volumes. Like a traditional microarray, it's on the order of microliters, but you do lose your sample after that. We can actually recover that sample, and it's not wasted. We're able to identify that first round of tests, but maybe the need is to do something other than sequencing or something other than that fluorescent readout. You do have that option to kind of continue on and use that sample elsewhere. Thank you.
Excellent, thank you. And I have a question I think this applies to anybody on the panel. Comes in from Mike for VR on YouTube. What are some of the biggest challenges you've encountered since you began biodetection programs in 2018? How have you adapted and modified your testing to overcome those challenges?
One unanticipated challenge that we encountered on the Felix program.
Thank you.
Sorry, we're trying to minimize background noise.
For what it's worth, it doesn't come in too much, but yeah. Sorry, guys.
So thank you for that question. One challenge that we encountered fairly late in the game on the Felix program, as we moved to more complex samples containing DNA from plants and mammals that tend to have much larger genomes than pathogens, is just the raw computational power that's required to sift through the millions of reads, of genetic reads that you get from the sequencer to make sense of what is natural versus engineered. And I mean, we had a couple of pivots during execution on the Felix program. One of them was to prioritize the computational tools to be more readily scalable. I mean, one of the features of Felix computational tools is that they scale with the computational resources you have available. And so if you are the federal government and you're only limited in how many servers you can requisition in the cloud, Felix is a game changer, right? You no longer need to hire 1,000 analysts to manually sift through the sequencing data. You can just requisition 1,000 servers and be done in a couple of hours. So we've for the most part addressed that pain point of the computational scaling to make the tools more efficient. I will mention one other pain point that we encountered during execution, which was that at this point we're fundamentally limited by the quality and the volume of training data that's available to train the computational tools. In particular, as you move to more subtle forms of engineering, like single base edits, if you don't have a large enough training database of samples that are what are called wild type, which means they're not engineered, they're naturally occurring in the wild, where you're able to understand what natural variation looks like at the level of single random mutations, and to be able to distinguish that from deliberate targeted engineering at the single base level, this becomes a very difficult problem. And so one of the lessons learned from the Felix program is now we know what we need to prioritize. We need to prioritize gathering and curating more diverse and better quality training data of engineered versus wild type organisms. And that's actionable by the federal government, by private industry, by philanthropies that want to contribute to the biosecurity space. It's a low-hanging fruit for impact, for getting us from 70% detection up to 100% detection. And I'm happy to turn this over to others who have thoughts.
I can answer from a wholly different extreme. So I really love this question, and specifically, you mentioned 2018, right? So the other half of this problem, which is about building baseline data, it's about how do you build large-scale collection of samples, and how do you do that in a way that is respectful of privacy? How do you do that in a way that's respectful of the genetic data that is non-human being collected, right? And so we've run school programs. This was the COVID experience of like, how do you build this infrastructure in a way that is aligned with values, and we care very deeply about that. So I think one of the things that we've been very clear about is how do you build this baseline data, making sure people understand that this is about non-human genomics, this is about pathogens, this is about bacteria. And it's a communication challenge like everything else in public health. And so for the last 3 years, I think the whole community has learned a lot about how to talk about these topics, how to engage with communities at its height. We're in 4,000 schools across the country collecting samples to look for COVID, yes, but making sure that parents and families were comfortable with that. And David, I think to build that baseline data, we have to find these ways, whether it's in our communities or obviously in places like farms and rivers. There's different kind of learnings we have about how to collect that baseline data in a way that is then kind of appropriate for this new domain, which is very complicated, and we have to, as practitioners, do a really great job thinking about how to communicate that to the entire world, really.
Thanks so much, everybody. I had another question come in from Kenny Dwyer on YouTube, who asked, what has global demand been like for this, and how do you expect that demand to change in the future? Many believe biosecurity is a COVID-only application.
So I have thoughts.
But it's not COVID only. That's for sure.
It's not. COVID underscored the profound societal value that Felix biodetection tools are going to have for us. But this program is about rapidly detecting and mitigating the next biothreat. right? As with everything that the intelligence community does, we're not just trying to understand what's happening today, we are trying to understand what's going to happen in the future, what could happen, and how we can either prevent or mitigate its impact. I will say that we have had robust demand for the Felix platforms, all 6 of them. Not, I mean, we are extremely grateful to be sitting here with 2 of our very high-performing contractors on the Felix program today, but we have had robust demand for all of our Felix platforms from partners not just in the United States, but in the usual partner countries that the U.S. intelligence community works with, and I'll stop there. And so I personally, not speaking for Dr. Marsh or for IARPA, I personally think that we will all be safer if these tools, this new category of tool that we have created through the Felix program, which did not exist before, became broadly available to anybody that wants to use it. Not just the United States, not just Five Eyes partners, but any country. Just having, I mean, it's a deterrent for starters. If any would-be bad actor, just by virtue of knowing that the tools for rapidly detecting what they're trying to do are out there, might think twice. It's in the interest of public health to be able to arm developing nations with the tools to rapidly detect emerging pathogens that might be spreading to humans through zoonotic transmission. So those are my opinions on the subject. I'm expecting that there will be worldwide demand for these types of tools in the future. And we're here today to tell the good news story that we have these tools available for everyone.
I just want to foot stomp that. And, you know, we started this program back in 2017. Nobody even thought about COVID, right? But we understood the threat that bio presented to the world. And so we knew to get ahead of it with that automated detection and to develop these platforms that now are here, right? It didn't exist before. Thank you to all you guys and what you've done, really foot stomping how important that is. But, you know, COVID is the incident that brought bio to the forefront and made us recognize something that we actually understood was a problem well before anyone else did. So I think that to tuck it away in a corner would be the wrong thing. It's really getting it out there so everybody's got it and understands, because that reduces the threat that's then out there. So that's my 2 cents.
Yeah, and I would point to IARPA's leadership here, not just in getting on this early, but also in making it available and awareness of this to other folks out in the world. And thank you for saying the point about this being available globally as being essential. I mean, I think this is the reality. You know, it's very similar to cyber, right? You know, it doesn't really matter where that computer virus pops up in the world. It can end up on your computer here or in another country, and there's a shared challenge. And so that opens the door to these tools that then, as David's saying, leads to deterrence. And I think that's critical. So really, again, yeah, thank you for that leadership.
Brilliant. Thank you so much. I have a concentric specific question from JR. I'm going to bundle them together. They ask, how can this be integrated into Concentric's wastewater surveillance product? Also, were these tools ready to use in 2020 at the beginning of the COVID pandemic? If so, can you talk about how they were used?
I'm going to give the wastewater question to Josh.
Can you repeat the waste?
Of course, of course. So how could this, I assume, meaning Endar, be integrated into Concentric's wastewater surveillance product?
All right, absolutely. So, you know, in Concentric, we're building, you know, a full chain from distributed sequencing, you know, sample collection, sequencing, and analysis packages. And so Endar is one tool of many in an arsenal to identify engineered, you know, threats in wastewater sequencing. To get back to the point on was this used in 2020, at that phase, we were just entering from phase 1, or transitioning from phase 1 of the program, where we were trying lots of approaches and seeing what fit, into phase 2, where we were really building a transitionable final product. And so, you know, at that point, we were, you know, using Endar in the sense of testing and evaluating it on a variety of applications. We were not deploying it on things like wastewater at that time.
And I can just add on the wastewater specific program, I think the mindset to think about is We're collecting wastewater from building level wastewater, from municipal wastewater sites, from airplanes. And so this tool, other tools that are being developed, the idea is that that's a medium of collection, and we should be constantly, obviously with each of those, there's partners and their decisions are going to drive how you deploy these technologies, but absolutely is the roadmap to detect for engineered items in those samples, but also other types of pathogens, other types of variants, other types of threats. And it's thinking about it like a package of analytics tools. It's very much how you should think about wastewater or other types of collection combined with these types of analytic tools.
So let's talk about Felix and COVID for the latter half of that question. So the latter half of the question was, what was Felix's role in when the COVID pandemic began? And this is one of the things that I am most proud of from my time in government service. It's probably the fastest impact any project I've ever worked on has contributed for the public. And it's because of IARPA, it's because of our excellent research teams and their commitment to rapidly pivoting our tools to an emerging threat to inform policy. Felix started, we announced the program in 2017. When the first genome sequence of the SARS-CoV-2 virus was publicly released in January of 2020, Felix tools were primed and ready to make sense of it. It took a nominal amount of retraining of our computational platforms on a broad spectrum of viral genomics data in order to retrain the platforms to make sense of the novel pathogen. And it took 8 minutes of analysis for one of our Felix platforms from the Broad Institute at MIT to give us a very detailed analysis of taxonomically where this new beta coronavirus fits in the spectrum of known bat coronaviruses and what parts of it were meaningfully different, how that relates to known natural variation in these pathogens, and to tell us quite clearly and quite quickly in January of 2020 that we didn't see anything consistent with deliberate engineering in this organism. And so IARPA instigated that analysis. It was supported by our performers on the Felix program. We reported that up to the Director of National Intelligence and notified congressional oversight. And in April of 2020, the Director of National Intelligence put out a rare press release affirming the IC's findings that this was not an engineered organism. And that helped to counter misinformation. Within a span of a couple of months, that really helped to change the narrative, I think, within government and in some other quarters about how we should appropriately proceed, how we should think about and how policymakers should proceed. And I'm happy to pass it to Catherine if she has anything else. So hopefully, I am so proud of the work that IARPA and our performers did on the Felix program to enable that policy impact for the federal government so quickly. It might even be the fastest impact that any work in the biotechnology domain has ever had on the government.
Thank you so much. I'll remind anybody watching this stream that they could submit questions in the chat. Our next question comes from Ethan V, who asks, and I assume this refers to both technologies, could the technologies be used as an omnibus test for diagnosis of different viral and bacterial infections?
That's not something we've specifically worked on. It seems like one application that could work. I guess the thing that we've been working on that's most similar to that is actually on this kind of wastewater monitoring thing, where we've been doing an internally funded project recently trying to use this type of hybridization approach to look for many different viruses and how you could evolve and understand new strains that are coming in and what all is going on there. So that's kind of the closest thing that we've worked on to that.
And similarly for us, the focus of the current version of Endar is really looking for genetically engineered biothreats. And so, right now, to my knowledge, there are very few genetically engineered pathogens, although this is something that could conceivably happen and that we would want to build towards in future versions. At present, there are components of Endar that help you figure out, what species are present in a sample. And so there is some kind of baseline there that could be built for something like a testing application. But right now, it's not in a state to make an omnibus test.
Excellent. Thank you so much. Our next question comes from Angel, who asks, considering the climate change challenges we have experienced over the years, is there any contribution that these technologies can make towards the reduction of greenhouse gas emissions? It's a very interesting question.
Yeah, I mean, one of the things so this goes back to the sort of baseline comment, right? So how can synthetic biology be applied around climate response, period, right? Like, well, the way to think about this is cells basically run on digital code in the form of DNA. One of the reasons we were able to perform well on this project is Ginkgo has designed more synthetic DNA than anyone in the world, and so we have a great data set on the engineered side. But, as we spoke about, you also need that data set on the naturally occurring side. You want to get that big training data. Well, that has utility for biosecurity. It also has a lot of utility because the way that we actually go and engineer a cell to do something new in the world is we go learn from nature and pick a tool out of biology and put it to work for a human application. So to give you a specific example, we have a project with Bayer Crop Sciences to engineer microbes to pull nitrogen out of the air and fertilize crops. Well, we didn't invent how biology does that. That's already how legumes work in the roots of soy. Remember crop rotation and all that in your garden, right? Like the microbes in the roots of legumes have genetic code for producing fertilizer without chemical synthesis of nitrogen fertilizer. Well, corn and wheat, they don't have those microbes. So we have a project to use the DNA code that we learned from nature in the legume microbes and put it into microbes that would live on corn. That could come out of that baseline non-human genome sequencing. We could get many genetic tools like that, and inevitably some of them will be relevant to climate because as far as I'm concerned, biology is the only manufacturing technology that actually knows how to produce things in a sustainable way on this planet, right? It's been doing it for 4 billion years, and we should just go and humbly learn from it, and this would be a great way to do that.
Fantastic. Our next question comes from Kenny Dwyer, who asks, are there any ethical issues in collecting DNA samples from the public domain? It's a great question.
One thing I want to emphasize is that at no time did the Felix program collect or use human DNA. IARPA takes the protections of the American public and humans in general very seriously. We have very stringent protocols in place on our programs where if you want to, or if there's even the possibility of using human DNA or human anything, there are so many administrative hoops and approvals that one needs to jump through. It was not a focus of the Felix program to detect engineered humans. I think that's taking that question to its logical extreme. We don't have that capability currently because we haven't trained on human data. And we're not going to. Yeah. Does anybody else have anything?
Surely, to David's point, the civil liberties and privacy protection that we have in place on every single program at IARPA and ARPA and the other parts really take that into account up front. We don't want to go down those roads. That's not what we're here to do. We're understanding of the science, right, and how to go forward and be able to detect that engineering protocols and what's changed. So.
It also should be applied to the non-human genetic resources. So I think there's an opportunity globally for countries to think more about the biology within their borders as a resource. And we should be making sure we're equitably rewarding countries that have a lot of biodiversity if that biology is used commercially, for example. And so there's a whole line, there's a lot of good, there's a lot of international treaties, a lot of discussion about this. Digital sequence information around Nagoya would be the thing to Google if you want to get a sense of where that world is heading. But I think it's a really important point in the long run for the industry. But yeah, so just to flag that too. And the human stuff, I think, is the much more radioactive ethical topic, but the non-human genome sequences have a component as well.
Excellent. Thank you so much. Our next question comes from Red Panda, who asks, it's a hypothetical, let's say a harmful strain was detected. How fast would the public know about it? How confident can you be in the detection? Any false positives would, of course, induce fear and lose trust from the general public.
I've never gotten a question from a panda before, and
we're used to it
this
here.
has made my morning. So the intelligence community does its work quietly and in the background, right? I said before, when we're doing our job well, you'll never hear about it. So from a government-led biosurveillance standpoint, I think it would be at the discretion of the president when the public would be informed about a novel threat. However, as I've said before during this briefing, I personally would like to see these new biodetection tools in the hands of anybody around the world who wants them, who has a use case for them, whether it's governments, whether it's nonprofits, right? NGOs who are in the business of biosurveillance to try and protect people in developing nations or elsewhere. Possibly even individuals who want to have an impact in their communities. And I think that if you see the broad adoption of Felix experimental tools, I mean, if you can just hold up the mini microarray, you saw in the demo earlier how easy it is to use these tools for rapid detection. And they have a handheld, battery-powered device that you can use to fluorescently identify if there's a detection within a span of, I don't know, minutes to an hour. This is something that could be used by anybody in the field. And so if there is an emerging pathogen, if there is engineering of concern that's out in the world, and people have access to these tools, I'd like to think that people would know quickly and that there's value in letting people know quickly. I think that how we react to a pandemic that is caused deliberately should be differently than we react to a pandemic that is caused naturally. So.
All right, so just to add to that, this is like a very important question of aspiration as much as anything else, and I think there's two really important models to think about. When we talk about the technology roadmap for development of these tools for collection platforms all over the world to detect threats, to be able to collect and then detect threats, it is with an ever pushing mindset towards real-time monitoring. All of those words matter. Real-time monitoring. We are not there today, but we are there in other things, right? Think about hurricane monitoring. At one point, Jason's from the east coast of Florida. I don't know if your ancestors were, but like there's storm clouds on the horizon. That's like, is it a hurricane or is it something else? And then it was buoys, and then it's satellites, and it's prediction, right? So that is technology roadmaps. And remember, like at one point, monitoring hurricanes from satellites must have been mind-blowing science. But today it's like, of course, why don't we do that? Same thing with cyber, right? Like I was born in 1983. My first computer, if you remember when we had Windows 95, no offense, Microsoft, but like as soon as there's just like a buildup of the number of viruses on your computer, and then literally you just had to throw it away and get a new one. That was like the state of the art in antivirus, malware protection software. Now, like once a year, you read on CNN, like quickly patch your iPhone. The rest of it's just happening behind the scenes. Technology development, it's monitored globally, constantly patched, constantly updated, because it's real-time monitoring, right? So I think we have to have the same aspiration with real-time monitoring as it becomes with ATCs and Gs, with DNA and RNA of dangerous biology, and we'll get there, right? And we'll get there with partnership between the private sector, government, technology development, and some number of years from now, it will look exactly the same in mindset if we get it right. And so I think that's like the mindset to think about it.
Which would be a huge public health boon, right? I cannot emphasize what is really exciting to me about this is what I just want to highlight that what Matt just said would be a huge public health boon, right? So what that looks like to have the ability to detect, respond, patch a malicious piece of genetic code that was developed by someone who's being a bad actor on the international scene looks a lot like shooting down a natural virus, which is, boy, would that be good for the world, right? And boy, are we still exposed to it right now, as evidenced by COVID-19. And so I really think these tools have an enormous immediate both dual use and public health opportunity as you think about global surveillance, with obviously the emphasis today being on maluse. But what you're going to need to make this work, all that collection infrastructure is going to have immediate dual use public health opportunities.
I have a question from Jose Antonio in the chat who asks, maybe you already answered the question, if so he apologizes. Is it possible to distinguish engineered SNPs from natural mutations, so single nucleotide polymorphisms here?
This was something that thank you for that question. This was something I tried to touch on in my earlier remarks about distinguishing natural variation from subtle single base engineering. While I can't comment on the specific capabilities of all of our platforms that we've developed on Felix, I did highlight it before that is a challenge that we highlighted through this program. That in order to have that capability of reliably, confidently distinguishing targeted single base edits from naturally occurring single base mutations, we would benefit from more diverse and deeper training data, right? So to go out and take, bat coronaviruses as a motivational example, it's a controversial issue that somebody might have been out collecting and studying bat coronaviruses. That's exactly what we need in order to deepen our understanding of what natural variation looks like in pathogens, so that we can tell when somebody has modified it deliberately versus this has emerged naturally. And we will never be able to answer the question about whether there is a bad actor who is engineering pathogens to do harm versus a natural zoonotic transmission until we have that deeper understanding of variation in the natural world. So hopefully that answers your question.
That's great, thank you. And the last question will end here. I'm sure folks have places to be. It's from JR, it's multi-part. He asks, do you expect there to be future collaboration between Ginkgo, Draper, and IARPA? Are there any other big problems to solve, or is it maintenance work from here? Also, can you talk a little bit about machine learning advances that occurred between 2018 and 2022, or even just 2020, and how they tangibly moved the project forward? So, questions about collaboration, about machine learning.
I'll give my response first, and I would love to get responses from the entire panel. IARPA selected Ginkgo Bioworks and Draper Laboratory for this briefing, specifically because of their excellent performance on the Felix program, on the extremely difficult evaluation that IARPA conducted to really surface the limits of our current detection capabilities, which are extraordinary. We also selected these teams for this briefing because of their demonstrated commitment to continued support and further development of their biodetection platforms. We're not in the business at IARPA of delivering one-offs, right? We don't just want to demonstrate that something is possible and then we move on to the next problem. We want to develop a sustainable and robust ecosystem of companies in this space that can sustainably provide the services that the federal government needs to support its biosecurity needs. And I just want to applaud our partners who are here today because they have shown us that this is a long-term priority for them.
So we do high-risk, high-payoff research. Everything is full and open competition. So if we have hard problems, we go out there and we don't presuppose a solution, right? We go out with a challenge and look for great ideas that we want to invest in to de-risk and to take a variety of approaches that we're going to look at for the community. So this was a great endeavor, and we've been engaged. We don't do that maintenance kind of stuff at IARPA. We're going to transition it on to our IC partners. Our IC partners are going to take it and they're going to operationalize it, deploy it, use it. Okay, that's not where we turn it over. And so, as David said, we have turned over the artifacts from this program to a large number of IC and international partners that'll keep going with that. In terms of the machine learning, you know, advanced compute and machine learning along the way absolutely has made a difference. But as with all artificial intelligence, machine learning efforts across the community writ large, it's all limited by curated data. Having great data, that's a unifying theme I hear on program after program after program. And so those having, it's not just having training data, it's having well curated training data that you can use to be smart. And then how do we get smarter about not only using that, but being able to use less of it or more targeted of it to learn and further develop. So do I think that we would hold them in great, yes, if they made a great proposal? I can't guarantee anything. That's fair.
Yeah, and I just echo some of what Matt said earlier, but since the mic's closer, I'll take it. Ginkgo is committed to biosecurity long term. You know, as I said, I think we're entering a DNA age, like the electronics age. You know, the next 50 years are going to be about programming cells, like we today program computers by changing their DNA. You know, Ginkgo intends to be the largest platform for doing that, and if we want to do that, we need to do it with care. It's important technology, and as far as we're concerned, a functioning global biosecurity system is needed to develop this technology with care, and so we're there for it. And we'll be there for the U.S. government. We'd love to work with those partners, and also internationally, as was mentioned, absolutely. So yeah, we're committed.
Brilliant. I'd love to leave the last word in your hands, and if not, we'll thank everybody that watched this stream. Thank you for taking the time. We're always open to more questions about this. Please, we put out a blog and a press release about this today. Please read. Please join us again. Thanks, guys. Anyone have any final words?
Thank you.
Yeah, thank you. Yeah,
it was great.
Celebration to the early commitment from IARPA. I think this is the magic of the federal government stepping up, and it's awesome to see.
Cool.
Thanks so much, guys.